It's all yours. Okay, so can you tell me about the talk that you just gave?
So we're investigating the relevance, clinical relevance of intratumour heterogeneity, which refers to sort of diversity within individual tumours, focusing on renal cancer, this talk, but we're also doing quite a lot of work in lung cancer as well. Essentially what we're finding is that tumours evolve predominantly in a branched evolutionary fashion, which means that you've got spatial separation of subclones in kidney cancer. And we can begin to elucidate the clonal origins of the disease through this type of work and determine that the common clonal founding event present in every region in the 10 sporadic cases we've looked at is an activation of VHL, as one might expect, together with subclonal, that is, branched events that occur in some cells, but some subclones are not others, and some regions but not others. That also have their own driver events as well that evade detection with a single biopsy.
Okay, so are we looking at 10 plus types of tumour like we found in breast cancer?
I think every tumour is going to have its own selection of driver events. No two tumours are going to be identical. Each phylogenetic tree, each tree that we identify on a per patient basis is distinct and a little bit like a patient's own fingerprint, different from a patient with the same histological subtype of disease. But despite that, there are similarities, and the similarities are common clonal event, VHL loss, and of course many of the drivers are shared between patients, but they may occur in different orders or in different regions of the tumour. We don't yet know how many driver events are operating or are present in a single clear cell carcinoma of the kidney. Some of these tumours can be very big indeed, and we're still massively undersampling these tumours. So we don't yet know what the order of events, of driver events is within these tumours, and how many driver events are occurring in an individual tumour, and that's the subject of the sort of next set of experiments we're taking on now.
Okay, so yeah, what do we do next?
Well, I think with two things, a clinical aspect of this, how do we use this data to leverage our understanding of drug development in general? I think first and foremost we've got better approaches to target the trunk events in tumours. Would be a worthwhile strategy. So in the context of clear cell carcinoma of the kidney, it's VHL inactivation. So we obviously need better drugs that target that loss of function event. But also understanding cancer evolution is important. Thinking about this branched evolution in drug development terms means that we need to understand which branches are being selected for through treatment and which ones are being pruned and lost through treatment to get a handle on what's driving resistance. And then, of course, we need to think about how to target heterogeneity itself. And one aspect of this we're very interested in is immunotherapy, using the body's own immune system to combat the diversity we're seeing in these tumours. And then the second sort of more preclinical aspect of all of this is designing better models to mirror the human disease and understanding the drivers of diversity in these tumours, what's actually driving the spatial segregation of subclones that we're seeing in kidney cancer.
Okay, thank you very much.
Pleasure.
Anything key you'd like to add on to that?
I mean, I think the sort of the final aspects of this is really more generally we need to understand the two really key Darwinian facts of cancer evolution. What's driving diversity in these tumours and optimising selection pressures in tumours to essentially prolong progression-free survivals and hopefully improve overall survival in drug development.
Great, thank you very much.
Thank you.
Give us your name and title.
Oh, that's okay, don't worry.
Oh, yeah,
Okay.